me just give a brief introduction. Jerelle comes from Dominica. She did her PhD in Cambridge with David Wales, and she's there currently with Rosanna doing her postdoc, and she's also a fellow in King's College, and she's really the rising star of the field. So I'm very excited to see her speak, actually, for the first time. I cannot believe it. And if you're not following her on Twitter, you really should. So Jerelle, please, the floor is yours.
Thank you very much, Angela, for this very generous introduction. Can everybody hear me and see my slides good? Okay. So good afternoon to everyone. Thank you for the opportunity to share my research with you today. In this talk, I will discuss the formation of protein condensates in different salt regimes, and we'll describe what we have learned from both in vitro experiments and multi-scale modeling about the different molecular interactions that sustain these condensates. Now, the previous speakers have given very good introductions to the field, but I would start off by again highlighting the central role of protein multivalency in driving phase transitions. And as we know, proteins that encode multiple interaction sites are often described in the literature as being multivalent. And these interaction sites we speak of may take the form of amino acid residues, short linear motifs, or the stuff that Jeremy just showed us, or protein domains. And these motifs are generally described by Papo and others, as mentioned in the previous talk, as stickers. And when we have multiple complementary interactions sites or stickers that occur within a single protein, these stickers allow the protein in question to participate in homotypic interactions. So wherein multiple copies of the same protein can interact with each other in a favorable manner. And as we would be aware by now, that under certain conditions, these interactions can lead to the formation of percolating networks that sustain phase-separated liquid-like compartments. Hence, multivalency is a critical role in protein liquid-liquid phase separation and biomolecular phase behavior in general. And at a fundamental level, we really want to elucidate what are the precise molecular details of these interactions, commonly referred to as the molecular grammar of phase separation, and determine how this language of biomolecular phase behavior transforms under different environmental conditions. So in terms of the molecular language of phase separation, so far we know that protein condensation can be driven by hydrophobic as well as electrostatic interactions, including charge-charge interactions, cation-pi, dipole-dipole, and pi-pi stacking interactions. But more importantly, we also know that these interactions can be strongly modulated in different environmental conditions, such as if we change salt, pH, and temperature. And recently, experimental and theoretical work have predicted reentrant phase separation for proteins that undergo liquid-liquid phase separation via homotypic interactions. So for example, it was predicted computationally that certain proteins demix into two liquid phases at low physiological temperatures. And then if the temperature is sufficiently high, these proteins form a single phase. However, upon further increase in the temperature, the system can, some of these proteins or polymers can re-enter the phase separated regime. And similar behaviors have been predicted by others for changes in pH and pressure. Now, based on these predictions, we therefore question, can the monotonic variation in salt also lead to reentrant phase behavior in protein systems, particularly for those systems that are stabilized by homotypic interactions? So where you do not need an additional component as the systems that was just presented to us by Jeremy. So can these systems undergo reentrant phase behavior? And to answer this question, our experimental collaborators in the Department of Chemistry here in Cambridge in the NOS lab, as well as experimentalists in Albert C. Hyman and St. George Hislop labs, they probed the behavior of fused in sarcoma protein which is commonly known as FUS, in different salt regimes. And these experiments were led by Tim and Georg here in the Department of Chemistry in Cambridge. And what they found was interesting. So they found that up to concentrations of about 1.5 millimolar potassium chloride, the protein forms phase-separated droplets. And from about 1.5 millimolar to 1.5 molar, what is observed is that the system forms a well-mixed phase. Surprisingly, above 1.5 molar, the system re-enters the phase-separated regime. And importantly, condensate formation in the high salt regime is fully reversible. So adjusting the salt concentration back to the intermediate regime yields a well-mixed phase again. And it is also important to note that condensates are observed at the usual low micromolar protein concentrations that are found in cells, as well as at physiological temperature and pH values. And so the next obvious question is, how general is this behavior? Do we see this behavior for other systems? And in fact, we do. So in addition to FUS, what was observed in the in vitro experiments was that salt-induced reentrant phase behavior is observed for several other proteins, including the pathological mutant of FUS, TDP43, BRD4, SOX2, and annexin A11. All these systems undergo phase separation by homotypic interactions at low salt, as well as at high salt. And interestingly, what they found was that the high salt condensates have similar size distributions and shapes as their low salt counterparts. And so macroscopically, these condensates are more or less the same. They're indistinguishable from each other. Now, so these observations beg the question, what are the molecular interactions that lead to the formation of condensates in this high salt regime? And more importantly, are these interactions the same as those that sustain condensates at low salt? Now, from our biochemistry and our chemistry, we know that significant drop in protein solubility upon addition of salt, which is commonly referred to as the salting out effect, has been attributed to hydrophobic effects. And so one of the leading hypotheses in this project was that enhanced hydrophobic interactions coupled with weakened ionic interactions may be the key drivers of protein reentrant phase separation in this high salt regime. So to probe whether this was in fact the case, on the experimental side, what was done is that our collaborators selected compounds that were known to act as disruptors of electrostatic as well as hydrophobic interactions, and tested the ability of these disruptors to dissociate preformed FUS condensates in different salt regimes. And what they found was that at low salt, all of the disruptors, so both your electrostatic as well as hydrophobic disruptors, were able to dissolve FUS condensates. However, at high salt, only the hydrophobic disruptor was able to dissolve the condensate, and your ATP and polyuridine RNA, which are acting as electrostatic disruptors, did not show any observable effects, suggesting that at high salt, where electrostatic interactions are screened out, phase separation may primarily be a hydrophobically driven process. And finally, in the in vitro experiments, or the final set of experiments that I will show you here today, what was tested was the ability of the high salt conditions to now induce phase separation of proteins that do not phase separate under physiological salt. So a good example is PR25, which is made up of a lot of arginine residues, and at low salt, it does not undergo phase separation by homotypic interactions, because in that regime, its homotypic interactions are dominated by arginine-arginine repulsion. However, at high salt, the system does undergo phase separation on its own. And again, we also found that whereas the hydrophobic disruptor was able to dissolve the condensate at low salt, at high salt, the electrostatic disruptors did not show any observable effects, again suggesting that liquid-liquid phase separation in that regime may primarily be a hydrophobically driven process. Now, armed with these bits and pieces of information from the experimental work, we wanted to get further insight into which interactions are really important. So in terms of the amino acid sequence, which interactions may actually be important for driving this reentrant phase separation behavior. And to study this computationally, we use multi-scale simulations. So in our simulations, we combine two complementary levels of resolution. First, at atomistic resolution, we investigated how the relative contributions of electrostatic and hydrophobic interactions among pairs of amino acids change as a function of salt. And then we use residue level coarse grain simulations to probe whether the changes that we observed in our atomistic simulations were consistent with protein reentrant phase behavior. So on the atomistic side, we first selected pairs of amino acids that can establish the various types of molecular interactions that we're interested in, or that may be important for protein phase separation. And we then probe the interaction between these amino acid pairs using umbrella sampling, which is a well-established technique. So in umbrella sampling, independent molecular dynamic simulations are performed at different points along a reaction coordinate. And for each simulation, the distance between the pair of amino acids in question is restrained via a Bayesian harmonic potential. And what this potential allows us to do is to sample the interaction between the pairs of amino acids at a given distance or in a given window. And then for each window, we will obtain histograms of the visited configurations, which we can then combine to compute a potential of mean force or free energy for the interaction in question. And so using this approach, we're able to probe how the interaction strength between pairs of amino acids varies as a function of salt. What we found was that at short distances, the attractive interaction energies between oppositely charged amino acids, perhaps not surprisingly, decrease monotonically with increasing sodium chloride salt concentration. While interactions involving uncharged residues in general increase monotonically with salt. Now, importantly, we find that interactions between basic and aromatic residues, that is your standard cation pi interactions, exhibit a more complex electrostatic hydrophobic behavior, which yields considerable interaction strengths at both low and high salt. Now, more generally, and perhaps the most interesting part of the computational studies was that we find that the strongest attractive interactions are those where both amino acids in the pair have pi electrons in their side chains. So we believe that these pi interactions between the amino acids may actually be important for sustaining liquid-liquid phase separation at both high and low salt, and not just important, but dominate the interactions that are underlying these phase behaviors. And this dominant role of pi interaction is even more evident when we consider cases where both residues have the same charge. So the arginine that I referred to before in terms of in your PR25. So we see that where both amino acids have the same charge, but they both have pi electrons in their side chain. This pair shows a more or less repulsive interaction at low salt and a weak attractive pi attraction as you increase the salt concentration. And now when we put all of this together, these observations support the hypothesis that we had that at low salt, protein condensates are stabilized by a combination of electrostatic and non-ionic interactions, while at high salt, hydrophobic interactions may be the strongest, and in particular, pi interactions may dominate and may really govern the phase behavior of these systems in the different salt regimes. Now, finally, we wanted to see whether the changes that we saw in the atomistic simulations, were they consistent with the reentrant phase behavior that was observed in the in vitro experiments. And to recapture the salt-mediated reentrant phase behavior, we use residue level coarse grain simulations. So in the coarse grain model, each amino acid is represented by a single bead, and the energy of the system is computed as a sum of harmonic bond terms and electrostatic terms, which are computed using a Coulomb term with Debye-Hückel screening and your short-range pairwise hydrophobic contacts. And then we then tuned the electrostatic and hydrophobic parameters of the coarse grain model based on the trends that were captured in our atomistic simulations in order to mimic the scenarios of the different salt regimes. So to test whether the proteins phase separate within this framework, we use direct coexistence simulations. And this essentially involves simulating both liquid phases, so your condensed and your diluted phase, in the same simulation box. So we begin by setting the coarse grain model interaction parameters to those corresponding to the low salt regime. And we then prepare a system that contains multiple copies of the same protein, perform NVT ensemble and MD simulations. And then from that, we can determine whether the system is phase separated or not at a particular model temperature, and this phase separation would be marked by the presence of sharp, well-defined interfaces. And then we can repeat this at different temperatures to see what the phase diagram for the system at the low salt regime looks like. And then once we have the phase diagram for the low salt regime, what we do is that we select a case that is close to the critical temperature of the system, but below the critical temperature, because these systems display UCST behavior. And then from there, we can modulate the interactions, that is from low to medium to high salt, allowing the system to equilibrate in each case and to observe whether our changes in the model are consistent with the different scenarios observed experimentally. And long story short, what we found was that consistent with the experiments, we find that at low salt within the model, first undergoes phase separation. However, PR25 does not. Whereas when we scale down the charge-charge interactions based on our atomistic simulations, and as expected, we see that the first condensate melted, and again, in that regime, PR25 does not phase separate. And then finally, to recapitulate the reentrant phase behavior at high salt, we increase the strength of hydrophobic interactions, including cation pi pairs, by 10% in the first system and 30% in PR25, and while keeping the electrostatic interaction scale down. And these enhancements in hydrophobic interactions was sufficient to yield reentrant phase behavior for the first system and induced phase separation for PR25. Now, it is important to note that when we analyze the molecular organization and density of the condensates at low and high salt, these properties are very similar to each other. And so hence within this model, the system does in fact re-enter a previously encountered phase separated state. So collectively, our experiments and simulations reveal that certain proteins do exhibit a salt-dependent reentrant phase behavior, and that different molecular forces stabilize condensates in the high and low salt regimes. Now, to put this study within a broader perspective, the proteins shown here to undergo salt-mediated reentrant phase behavior are all essential for cellular and developmental processes. And importantly, some of them have been implicated in neurodegenerative diseases, such as FUS and TDP43 have been linked to ALS, polyglutamine disease, frontotemporal dementia. And thus the existence of this salt-induced reentrant phase for these proteins may hold important implications for not only the aberrant functions of these systems, but as well as the druggability of the related condensates. And from a more general perspective, the findings in this work add to a growing body of work suggesting that, yes, the forces that sustain phase-separated compartments may be are dictated by the amino acid sequence, but also may be critically influenced by the environment that the condensates are in. And so changes in environmental conditions can dramatically tune our phase boundaries, as well as the material properties of the ensuing condensates. And finally, an important hypothesis that stemmed from this work is that, and that is left to be investigated further, is that perhaps in the origins of life, the high salt concentrations of the oceans could have perhaps facilitated the formation of protocells via phase separation, possibly concentrating select biomolecules into these phase-separated compartments. And with that, I would like to thank you for listening. I would also like to thank my amazing colleagues and collaborators on this project, in particular the members of the Qualipado, Knowles, Alberti, Hyman, St. George Hislop Labs, as well as thank Jitain Mittal and Greg Dinon for their assistance in implementing the cost-grain model, and Papu, who offered useful discussions that greatly improve the quality of our work. And last but by no means least, I will not be here without having funding. So I would like to thank all the funding bodies that support our research and my opportunity to be able to do work that I'm very excited about. So thank you.
Great, thank you so much, Jerelle. This is excellent. So do we have any questions at this stage before we start with, I guess, a panel discussion? Can I ask, I mean, and I probably don't know the literature well, and I'm kind of superficial in reading papers, but I always, people often speak about the role of ATP in the formation and control of these aggregates. But actually, and I always had imagined this would be kind of an active process where there's some enzyme like eating up ATP and so on. But you're also saying that just its sheer high charge can play an important role in controlling these kind of finite size aggregates, and I guess the whole phase separation process is kind of that also the message that I could take from this.
Yes, so when we think about sort of framing the ability of these systems to form aggregates or to phase separate or not, and we think of a study like this where we see that essentially ionic screening is what is sort of driving this behavior, we can think of different scenarios inside of the cell that might not necessarily involve the addition of salt, but might involve the addition of post-translational modifications, for example, that might elicit ionic screenings or changes in the condensate environment that can drive it inside or outside of the phase-separated regime. So something that we sort of had to think about was that in the context of when you think about drug abilities and preventing these systems from forming this sort of irreversible condensates, when we think of therapies, we think of moving potentially harmful systems outside of the phase-separated regime because we think that they might continue to age and eventually form this pathological state. But because the environment that the system is in can cause it to even re-enter a phase-separated regime, we have to think critically as to how changes in the condensate environment can move it in and outside of a phase-separated regime and where we are in phase space. So these are some of the things that we have to sort of frame and think about in a broader context.
Thank you. Jeremy?
Yeah, so just a quick question about your system that you worked with there. In the condensed phase, how much space is there for water and counterions in these? How dense are your condensates?
Yes, so for the residue level cost grain simulations, we don't have explicit solvents and ions, right?
Yeah,
So,
but you can, but you have voids instead.
yes, yes. So typically you have, it depends on the system size, because you do not want to have the system interacting through the periodic boundaries. You can have as much as, well, not as much, but typically you may have as much as 3 times vapor versus your condensed phase. Or
No, I'm talking about inside the condensed phase. What's the volume fraction of protein inside the condensed phase?
So the volume fractions can vary from about 0.3, right, to all the way to very, very dense phases. So it depends on where you are on the
Okay, but 0.3 is at the low end, huh?
Yes.
0.3. Wow. Okay, that's much denser than I expected. All right, thanks.
Yeah.
Cool. Do you have any other questions for Jerelle specifically? Maybe if not, then I'll thank her and actually all 3 speakers from today. And now maybe we'll have just a brief discussion on several burning questions that we discussed prior to the start of the meeting. So maybe I'll post them here for just the ease of everyone. Oh, sorry, there's something. Am I allowed to share? Yeah, cool. Okay, here they are, burning questions. You can see that, right? So maybe I'll start with the first one that Jeremy seemed excited about, and it seems to be present, I guess, in the literature and also a bit trending on Twitter. Like, is every phase separation liquid phase separation? And nowadays, it's almost being used as a synonym. And if you kind of think you want to improve chances of your journal, you might get of your paper, you might get tempted to like throw in LLPS like randomly throughout the text. So I'd be very curious to see what our 3 panelists and actually any other person who's still with us, what do they think about this? So maybe Jeremy, if you want to start, you seemed excited about the controversy.
Okay, so my opinion on this is that, well, I'll go back to the famous saying that, all models are wrong, some models are useful. And, I think LLPS is an excellent model in order to understand what's going on in this system, in these systems. And, to make an analogy here, if you were to talk about, equilibrium binding constants in a cell, you can pick that apart easily. A cell is not an equilibrium environment. So why on earth are you using, chemical equilibria constants in order to understand what's bound and what's unbound? Well, it's very, very good most of the time. And unless you have some serious, kinetic constraints on it that push you way out of equilibrium, using equilibrium binding constants are an excellent model to understand what's going on in many contexts inside the cell. And I think the same thing is true for LLPS in a cell. LLPS is an excellent model for understanding how these structures are formed. Is it going to break down? Yes. These are extremely non-equilibrium environments, and you're always going to be able to push the model or the metaphor to the point where it breaks. I think that point where it breaks is, of course, going to be different and extremely interesting for each system. Is it the non-equilibrium features that cause it to break down? Is it the non-extensive features that cause it to break down? Is it, there's all kinds of ways that these things can fail to violate or fail to satisfy the strict requirements, but for LLPS. But like, for instance, it's recently been shown that paraspeckles are basically, micelle-like structures. And the interior of those micelles are stabilized by the same types of interactions that, if you just cut the tails off of these, RNA structures that form these perispeckles, you take them in vitro, they're gonna LLPS. So the types of interactions that drive LLPS are the same ones used to form these finite sized assemblies, in a perispeckle. So in a way, I think it's a little bit of an irrelevant question. Yeah, it's a pretty good model, and the model is going to break down in some really cool, interesting, and useful ways. And so understanding how it's valid and how it's not valid are equally interesting ways and useful ways of looking at the biology.
Mm. But what about, people often report, okay, we see stuff aggregating, and then they call it LLPS. Like, does it matter if they're liquid or like solid? Or does it matter if they're really phase separated or like finite size assembly?
Yeah. Well, that's a good question. So there's two answers to that. One is that, I think that these things, and there's lots of experiments that show this too, that they're going to look different depending on what length scale you look at it. I think they're going to look at solid on some length scales and liquid on the others. And this sort of, follows from the hierarchical arguments that I was making before. The other thing is that even if you don't have a hierarchical thing or hierarchical assembly, if you just look at, some of Rohit's, simulations, just basically really regular sticker and spacer things, these things are viscoelastic. So, characterizing whether it's liquid or solid, you've got to specify what time scale you're looking at there. So yeah, there's a point it breaks. If you're looking at, a solid aggregate, a protein crystal is not LLPS, but that's, is there a useful way to think about it in liquid? Is there a useful time scale at some processes happening that this thing is dynamic? If you can say such a thing, then yeah, probably the LLPS model is a useful way to think about your system.
Yeah, maybe, this is really a great question. Maybe I can add also the point about the size, right? Because that's also like another very interesting question, right? If you have, say, maybe tens of molecules or so interacting with each other, do you call that a liquid, for example, or so, or is it an aggregate or so? Probably they tend to be very dynamic also, so they have that kind of liquid-like feature in terms of, order or so, but still they wouldn't be called liquids also. So I think there are really a lot of like very interesting questions associated with size, time scale, etc. So yeah.
Any other comments on the first hot point? Cool. And then maybe we can touch upon, maybe David wants to say, please.
Just briefly, I completely agree on the viscoelasticity. It depends on time and length scale what you're looking at. What I find interesting, so that pertains to the liquid-liquid part of LLPS. But what I find more interesting currently is to think about what are signatures of phase separation in cell. So and that becomes also tricky to resolve when you go to smaller and smaller length scales, because thermal fluctuations basically wash out all signatures that you are used to from a thermodynamic system. And in some point, I find the discussion a bit weird because it's almost semantics. Physics doesn't care about whether you call something a droplet or phase separation or whatever. They're well-defined molecular interactions. And if you go down, thermal effects become more important. And it's more, the problem is more that we need language to describe what's going on. And I agree with Jeremy. If you formulate them all in that sense and it's helpful, then apparently on the parameter regime where the assumptions are useful and we can use this language. But I don't see what all this fuss is about, really.
I do agree. I do agree it's semantics, but then again, if you think about physics, which you said matters, might matter, right? Because when you go to small length scales where things are not as continuous as your model would like it to be, and where you have thermal fluctuations and so on. But also, I guess self-assembly started in the virus field, maybe, and I don't think you would call a virus assembly LPS, right? And yet again, I think it has some very similar features of things we do call LPS.
If we're going to make our cows spheres, do we really have the right to complain about somebody else's model?
No. But I think, it is helpful to discuss these things openly, and maybe for the first time, for me at least. So why is it so controversial? Why do people get so touchy about the topic?
I don't know. I always go back to sort of when I first started looking at proteins, and I was reading the literature about IDPs and globular proteins. When back in 1995, you started seeing more in the literature people talking about intrinsically disordered proteins. It was very controversial because at that point you didn't have the necessary experimental and computational techniques to really look at this very closely and to do it in a very robust and quantitative manner. And so people needed convincing. And now it is sort of, we're at the stage where it's the other way around, where we know that so much of the proteome is intrinsically disordered proteins, and now everybody is has well accepted that that is the case. Well, I don't think I've met anybody who hasn't, but I think for the field of liquid phase separation, in my opinion, there's still a lot of growth that needs to take place in terms of the development of experimental techniques, as well as the development of robust modeling toolkits, as well as the theory. And so because these things are now developing and people might not feel like they have a lot of numbers to back up what we're saying, then the controversy comes in. But I think once work continues to go out and you begin to get more evidence that these things actually do happen, I think the tides will turn. But with every sort of like shifting paradigm, I think people are going to have their different views. And again, because another thing is for when we think about phase separation of biomolecules, because it happens at such different length scales, there are so many different length scales that are involved in this whole process. We have, I think, a lot of work to do in terms of what length scale that we're looking at the system. And I really like that image that Jeremy showed where he was showing like the back in the 1970s, where you have this different studies showing that you do get this LLPS behavior, but it depends on where you're looking, right? And sort of like the environment. So one person can look at the same system and have, let's say, for example, magnesium ions present, and one person can look at the other system and not have magnesium ion system. And whereas one case might report a liquid, one person might report a gel. And I think those fine details matter, as well as developing the techniques. And once we just have to continue working at it, I think.
Yeah, I mean, I find it, shouldn't it be more controversial when things don't phase separate? I mean, you have so much stuff to sell that I didn't see why the fact that, I mean, is it surprising that things separate? I think maybe it's more related to the next question, is whether this separation is just kind of an observer, like it just happens, or whether it has a functional or indeed dysfunctional role, and probably sometimes it's either. So I would be curious to hear your opinions.
Well, I'll inject an opinion here that is mine and mine alone, and so therefore it might be a little bit controversial. But I think that, when you look at the dominant hypotheses for what these things do, most of them revolve around that there's some variant of a compartment. They're a box, and I don't think that that's what they're doing. I don't think that and there's some cases clearly where that is the case, but I think the vast majority of cases, they're not just boxes. And to tease a little bit of the work that Thomas and I have been doing, they actually don't work very well as boxes either. But that's still a story in progress. And I think a lot of the controversy is, are they functional or are they not functional, involves experiments where you either make the box or you don't make the box, and you ask what that changes to the function. And when you're doing that, you're by making this artificial system where you can turn them on and off, you're disrupting all the internal structure that actually allows them to perform whatever their actual function is. And so I don't trust those experiments. I think that they're more likely, these are not, the two states are not condensate or no condensate. These things have multiple internal states. And those internal states can be tuned by, whatever cell regulatory cycles we have, and we don't have the tools yet to see what those internal states are. And I think that's why the functionality has been controversial, is because we're not probing those internal functionalities of the condensates.
I think David had his hand up.
No, I just wanted to answer your earlier question. So I think it's not surprising at all that these things phase separate. If anything, I would expect the opposite is surprising, that there are no more droplets, because there's so many interacting molecules in the cell, and just from combinatorics alone, you would expect to lower the free energy by spatially segregating them. Dan Fengler has shown this very nicely in his work.
Yeah.
And so I think there's a lot of energy in the cell going into actually keeping it more homogeneous and more fluid-like, to counteract this thermodynamic tendency.
And how do you think the cell does it? Like, is it the sheer turnover of the things?
Turnover helps, definitely. Turnover helps. Forces might also help, right? Molecular motors, you have polymerization, all this.
Or local fluctuations, like active fluctuations due to,
I don't have a very good sense. It's more gut feeling, so to speak. But there are experiments that if you remove ATP from the cytosol, that it becomes more gel, solid-like, pointing in this direction. But then,
do you think that's just because you effectively lowered the temperature because you don't have so many, like, such high amplitude fluctuations?
I don't know.
Any other comments? Okay, so maybe the next one. Yeah, how much of quantitative matching do we have between modeling and experiments, especially in vivo? I'm not an expert on the vast literature that must exist on the topic, but maybe one of you has something to say. But maybe that's.
I think Rohit's group has done some really beautiful work on, quantitatively, matching phase diagrams and predicting, phase separation stuff from simulations. But yeah, I'm not sure. I would say that a lot of it is pretty qualitative at this point, but.
But maybe what Jerelle was saying, maybe we're missing also quantitative experiments. And that's a problem. Like, you can not do the matching if you don't have like solid experiments.
I would agree with that. Yeah.
And do you think we need like new set of tools to study this phenomenon, or do you think it's so attractive just because we can use all the tools we have already to study a newly discovered things? So is that why? Because I guess traditional soft matter has developed all these tools to study equilibrium, phase separation of polymers, and I guess dead matter, and now it's very attractive to apply it to living context. Or do you think we really need to develop something new to meaningfully address these questions?
Well, again, I'll interject here. I think that the really interesting things start showing up in these condensates when you start looking at wide ranges and length scales and energy scales. And when you talk about computational tools, that's something that the computation can frequently struggle at. And so learning how to integrate those different length and energy scales in order to see how these things function as a whole is going to be challenging. It's very doable, with the tools that we have. But yeah, that's what I see.
Yeah, but I guess, yeah, multi-scale question is such a universal problem for whatever you study. But then again, I guess it depends on the question you're asking, what kind of tools you'd need. And finally, yeah, I mean, there's a lot of mention of the role of phase separation in the design of new therapeutics. I mean, both, I guess, for targeting, but also for formulation. So do you have anything to add to wrap up the workshop on this topic?
Maybe I can add, say at least from my perspective, also as a kind of theorist. I mean, this is really a great opportunity, right? Maybe even adding to the tools, right? I mean, from a theoretical perspective, I think there are already quite a lot of theoretical tools that we already have from different areas, like traditional soft matter ideas and so on, but then also going beyond like control. For example, David is very interested in that aspect too. And so on, and still we don't know exactly how these principles apply to the system. So already kind of applying these tools will tell us a lot to the system where they apply to. And in the context of like therapeutics, well, I mean, like it's a big question, right? Because first of all, the design of compartments to kind of deliver drugs, for example, also. As a follow-up, for example, of my work, I've investigated what would happen if you have a therapeutic agent that would interact with the aggregation process, and there you would see, for example, an enhancement of inhibition, so reduction, for example, of the dosage that you could use for reducing, for example, the toxic effects or so. So that's actually a very attractive idea, and there are a lot of people that study the systems also from a synthetic perspective. For example, Paolo Arosio in Zurich also is kind of really trying to push this to the synthetic way also. So that's also kind of very nice from the formulation perspective. And then also the whole question about the aging of these droplets, which is, I think, a very, very important one, both in terms of understanding the mechanisms, which I think we are kind of starting to get an idea about, and then also thinking about strategies to kind of either, for example, revert the process or so. For example, this idea about salt is actually really interesting that Jerelle showed also, or kind of driving the inevitable, say, aging, maybe in kind of through different pathways that would be maybe less toxic also. So I think this is really a very, very, very big field and a big opportunity.
I would say that, if, your canonical model of a therapeutic is you develop an inhibitor that goes in and blocks some specific molecule. And, it's sort of a binary switch, you're on or you're off. And I think that, these condensates provide a whole different range of interactions in that they're tunable. You could go in and you could take a condensate and you can tune its property with molecular intervention. I could go in and I could, design a molecule that, makes the binding, blocks some of the binding sites and causes this thing to fluff up because the characteristic length scale between the binding sites is bigger. So I can, make the lattice spacing bigger, and I could, create more space inside there. Or I could do the opposite. I could, make shorter cross linkers that cause the thing to contract down. So it seems like we've got a lot of therapeutic knobs that we could turn on these things in order to modulate their properties. And it's not just an on-off switch. Is it active or is it not active? We can actually go in and we can tune the properties. And so in that sense, it seems like there's an awful lot we can do.
That's very actually and positive and optimistic. And it's, I think, a great opportunity to finish this workshop on such a note. So do we have any other final comments or questions for our excellent speakers today, and also all the other groups who joined? So if not, I think we should all thank Jeremy, Jerelle, and Thomas. Thanks so much for joining today. And we should also thank our audience who took time from their afternoon to come listen to the talks and also participate in the discussion. So thank you, everyone, and also Karen for organizing it. And Karen actually typed here in the chat where you will be able to find this event on YouTube. So in case you missed any